All right, so welcome back. So far, we have had two videos where we went over just a basic introduction of the cell, and we talked about the plasma membrane and cell walls that are found um, in many different types of cells. Now, we're going to focus on kind of the attributes of eukaryotic cells, specifically looking at organelles. And the first organelle we're going to focus on is the nucleus. The nucleus is the control center of the cell. Whenever I'm thinking about a cell, I tend to think about it as a working city. And I'll kind of use those examples as we kind of discuss these relationships. And so you can think of the nucleus as City Hall or a courthouse, or kind of like where legislations run and where all the instructions are kind of divvied out to the different people, the different businesses in the city, to kind of tell everybody how to work and how to function, and basically how the city is going to be ran. So that's basically what the nucleus is. It's the control center of the cell, and it holds all of the information, all of the directions for that cell in its DNA. And so the DNA is nucleotides, or I'm sorry, nucleic acids that are made up of nucleotides. So if you remember that from the organic molecules lecture. Now, each DNA strand, chromosome, has little stretches of DNA. So maybe it's, a couple hundred base nucleotides to several thousand nucleotides long. And those segments are a gene. And so in each gene, it provides specific coding, specific nucleotide sequences that is the information to make a protein. And so what the DNA does is it is transcribed into RNA, and then that RNA is translated into a protein. So the proteins that need to be made and how many of those proteins need to be made are all determined by the codons, or I'm sorry, the nucleotides that are in that DNA structure. And all of the DNA is held inside of the nucleus of the cell, so that control center. And so this is very much where all of that information is located for how that cell needs to function and work. And the proteins are kind of the workers that go out and do that work in the cell. And so the nucleus is separated from the rest of the cell, from the cytoplasm and all the other organelles, because it has a nuclear envelope. And this nuclear envelope is a double membrane structure. So there's basically two membranes, and it separates that DNA from everything else. But that nuclear membrane does have nuclear pores. So there are these openings, pores in the membrane that allows specific molecules and structures to pass from the nucleus into the cytoplasm. And that's actually how things like RNA will move from the nucleus into the cytoplasm, so it can be translated into proteins, which we'll talk about in the next video, or maybe this video. And so with the nucleus, like I said, we have these long DNA molecules that exist inside that nucleic structure, that nucleus structure. And these DNA molecules form what we call chromatin, and they form that chromatin by wrapping around proteins. And once they wrap around those proteins, they'll condense into these chromatin fibers. And one chromatin fiber, or a long chromatin fiber, is one chromosome. Okay? So for example, humans have 46 chromosomes. We have 23 pairs of chromosomes. So 23 times 2 copies of each one for humans. And then so the number of chromosomes for all species really differ. You have species that have like 4 chromosomes, and you have species that can have like over 100 chromosomes, just depending on what organism you're focusing on. Now, inside the nucleus, there's a place called the nucleolus. The nucleolus is this structure that is in the center of the nucleus, and it's where the components of the ribosomes are made. And then those ribosomes will exit the nucleolus through the pore and go out into the cytoplasm where those components of those ribosomes can be put together. So here we have a figure of the nucleus, which is the purple sphere structure. You can see the double membrane, the pores that exist inside that membrane structure. Then this purple kind of blob in the middle is the nucleolus, where those ribosomes are made. And this kind of violet, or I guess dark or purple, this would be violet, I guess, is another organelle that we'll talk about in the next video, which is called the endoplasmic reticulum. Now here is a TEM image of the surface of a nucleus, and then this is zoomed in to look at the individual pores that you see on the surface of that nucleus. Now the chromatin fibers, the DNA, exist in this kind of light purple space, light violet space here. And if we zoom in on a chromatin fiber, we can see that it's actually a DNA molecule that is wrapped around some proteins that are then kind of compacted together to make a chromosome. And so those chromatin fibers are DNA and proteins that again make one chromosome. And again, the number of chromosomes differ between the species of organism that you are studying. Now, so ribosomes are made in the nucleus, and the parts are then exported and put together in the cytoplasm or on the endoplasmic reticulum, where they will make proteins. And so ribosomes are very important for protein synthesis. And in eukaryotic cells, the components of the ribosome again are made in the nucleus and then transported into the cytoplasm. Things like the free-floating ribosomes, the ones that are in the cytoplasm themselves, will make proteins that remain in the cell to conduct their work in the cell. Then there are other ribosomes that will be attached to the endoplasmic reticulum. Those ribosomes will make proteins that will be packaged and modified and transported outside of the cell itself. So here we have an image of a ribosome, which is this kind of blue structure. Then some RNA. This is mRNA, so it's called messenger RNA, and we'll talk more about that when we go through the DNA chapter. And then here's a protein that's being translated. Okay. So this is kind of that ribosome structure that's translating RNA into a protein. All right, so here we have a picture of the endoplasmic reticulum, and then also the ribosomes that are kind of attached to that endoplasmic reticulum. So those tiny blue dots that are on the outer membrane of that ER is the ribosomes, and they'll make proteins again that will be transported and moved outside of the cell itself. And so one thing I want to talk about with kind of the transfer of information of DNA to a protein is that it undergoes a couple of different steps. So DNA has that has the base code, so those 4 bases, A's, T's, G's, and C's, right? The adenine, guanine, cytosine, and thymine. And those can code for RNA, and then the RNA can be translated into proteins. So what happens first is inside of the nucleus, the DNA will be transcribed into RNA, okay? And then once that RNA is transcribed and a couple of things are done to it, it's modified, it then goes out of the nucleus into the cytoplasm and then comes into contact with a ribosome and then that ribosome will translate it into a protein, just like we see in this image here. Okay, and so basically you have that kind of process here where you have the DNA making the messenger RNA based off of the information the DNA has. And then that RNA is moved out into the cytoplasm and then is translated into a protein, which is that worker, like structure that's going to go on and conduct the work of the cell. All right, so that's it for our nucleus video. We will pick up our discussion on eukaryotic organelles in the next video when we talk about the various structures that are part of the endomembrane system.